Stress causes fear, anxiety, frustration, and depression and can lead to a number of behavioral problems, including increased aggression, weight loss or gain, excessive grooming, barking, self-mutilation, or stereotypic behaviors.
Shelters provide a source of psychological stressors. These stressors include high level of novelty and noise, separation from previous attachment objects, social separation, unpredictable and uncontrollable environments, and disruption of habits. These stressors are known to activate the hypothalamic pituitary adrenal system, which is known to have physiological and behavioral effects on the dogs.
The hypothalamic pituitary adrenal axis is a complex set of direct influences and feedback interactions among the hypothalamus, pituitary gland, and adrenal gland. This intense communication network is a major part of the neuroendocrine system that controls the reactions to stress and regulates many body processes. In the brain, the hypothalamus is connected to the pituitary gland. The hypothalamus is stimulated by the sympathetic nervous system, causing a release of corticotropin-releasing factor, CRF. The CRF activates the pituitary gland to secrete adrenocorticotropic hormone, ACTH. The ACTH signal is received by the adrenal glands located on the top of the kidneys. ACTH stimulates the adrenal cortex to release cortisol.
Cortisol is recognized as a major indicator of response to stress. This response is similar across all species. In recent years, there has been increasing evidence that activation of the hypothalamic pituitary adrenal system has detrimental consequences for brain and behavior. In humans, it is hypothesized that stressors such as abandonment by an attachment figure may sensitize the hypothalamic pituitary adrenal axis so that the individual responds with a full stress response to progressively smaller amounts of stimulation. It is possible that abandonment of a pet at a shelter may cause a similar sensitization process. Cortisol can be measured in a variety of methods. The three most common methods include a plasma blood sample, a fecal sample, and a saliva sample.
In a study conducted by the Department of Psychology at Wright State University in Dayton, Ohio, plasma cortisol levels of dogs held in a county shelter for a number of days were compared to dogs maintained as house pets. 18 shelter dogs had blood samples taken every day that they remained in the shelter's care, and 17 pet dogs were sampled every day at the same time of day shelter dogs were. It was found that dogs at home have much lower levels of plasma cortisol than those of shelter. This study also showed that plasma cortisol concentrations of dogs for their first, second, or third day were much greater than those of dogs in shelters for 10 or more days.
Having higher cortisol levels in the first few days after arriving at an animal shelter has a significant impact on whether or not a dog is deemed adoptable. Healthy dogs that are not claimed by an owner are kept for an average of only 6 days before it is decided whether they would be placed for adoption or destroyed. So how can we help minimize stress of shelter dogs? Dr. Temple Grandin conducted a study in order to see if human interaction alleviated the stress response of dogs shortly after arriving to a public animal shelter. 51 dogs participated in 45 minutes of human interaction for 10 days, while 54 other dogs had no human contact. Salivary cortisol levels were examined from each dog on their second, third, fourth, and ninth day of housing. What they found was day 3 seemed to be the most stressful day for the dogs housed in the shelter, mainly because they had not begun to acclimate and are responding to consistently unpredictable surroundings. The data showed that on day 3, the contact animals had much lower cortisol levels than the no-contact animals. It was concluded that human interaction prevented sensitization of an endocrine response to novel situations. This study showed that interactions of humans may be an effective way to reduce the cortisol stress response of shelter dogs.
It is commonly believed that women possess a more nurturing manner and are more effective at comforting in times of stress. In another study conducted at a Wright State University, the influence of male and female petters on plasma cortisol and behaviors was observed. During their second or third day in public animal shelters, juvenile adult dogs had their blood drawn. The dogs were either not petted or were petted by either a man or a woman. 20 minutes later, a second blood sample was collected. The behavior of the dogs during the petting episode was recorded. The number of times that the dogs yawned and the number of seconds they spent panting was observed. Yawning was examined because it is thought to be a possible sign of displacement activity indicative of conflict, and panting because it is a possible sign of anxiety or fear. Data showed that having their first blood sample drawn resulted in higher cortisol levels 20 minutes later if the dog spent that time interval in a cage with no contact. This rise was prevented if dogs spent the 20 minutes being pet by humans. It also showed that dogs who were subjected to human contact showed less behavioral signs of anxiety or fear. However, this figure shows that men in the study were at least as effective as were the women in moderating the cortisol response level following the collection of the pre-test blood sample.
Overall, we are led to believe that elevated cortisol levels have a significant negative impact on the behavior of sheltered dogs, which causes them to exhibit characteristics that deem them less adoptable. These negative characteristics may not necessarily be the dog's normal behavior. It is important to minimize the stress during the early portion of an animal's stay at a shelter so that they are judged by their true personality and not by stress-induced behavior.
It seems that after hundreds of years of domestication, humans have acquired an immense social influence on the behavior and physiology of dogs. We have selected and bred largely for qualities as loyal companions. Human interaction represents a potentially powerful tool in trying to relieve and eliminate the distress of dogs in confinement.